So thank you very much, Padma, for this kind invitation, and thank you for having me to this nice place in Boston, to this conference, and giving me the opportunity to show you the perspective of a pathologist on copy number alterations. So first I would like to introduce myself. So I'm head of the Department of Neuropathology in Bonn, and this department houses a reference center for brain tumors. We mainly do second opinions in difficult cases for our colleagues, and we serve as the reference center for neuro-oncology studies, mostly national but also international studies, and we try to develop and validate new diagnostic tools for the diagnostics of brain tumors, and we also do education for residents as well as trained pathologists. We get about 2,000 new submissions for this review, and we classify the tumors according to the WHO classification of brain tumors. The last version is from 2007, and so this is a kind of language which is understood by all our pathology colleagues and also clinicians over the world. It is revised every 7 years, so I think next or the year after next year will be another revision on that. It's mostly morphological, but also other features are in there. So this reference center is not founded by me. It is existing since 1980 when Paul Kleijus founded that. He also contributed a lot to the first and second version of the classification, and then was taken over by Ottmar Wiesler in 1994 in Bonn, and since 2004 I'm heading this reference center. So what about copy number alterations in tumors? So there are some special findings in brain tumors which make it really interesting for diagnostic, prognostic, and predictive purposes. There are typical alterations in subtypes of tumors, which I'll show you some examples. One example, and I think Dr. South will get into this in more detail, is that in a fraction of very malignant tumors of adulthood called glioblastomas, these are very poor patients with short survival times. They have amplification of the wild type EGF receptor or of a mutated form, and this can be detected by this analysis. A second tumor, which is also present in young adults, is called oligodendrogliomas. These respond better to treatment, so the survival times are better. And the majority of these have a very interesting co-deletion. I will mention this in the end of my talk. So they lose both the short arm of chromosome 1 and chromosome 19, and this is a prognostic marker. And then in the pediatric field, and you shall know that children have relatively more brain tumors than adults. The most frequent one is the malignant medulloblastoma, highly aggressive tumor, and in this tumor it's now believed that there are at least 4 different subtypes with different copy number alterations. You can use it to make distinction between these biological different subtypes. And the last example is the second most frequent malignant tumor. It's ependymomas, which partially have a 1Q gain, which is a prognostic marker. So I will show you some of these examples. Now, what is the problem that we face as pathologists? We work with paraffin-embedded formalin-fixed tissues. We call it FFPE, as you know. And we frequently have only small amounts of material available. So we are restricted in DNA. Then the material may be contaminated by hemorrhages, by scar tissues, by normal tissues. So frequently we have to dissect it before we do any kind of analysis, not to be biased by normal cells. The tumor cells by itself could be heterogeneous, as you know, for some markers. So this is something we have to take into account. And the most problematic point is that as a consequence of fixation and embedding, the DNA is highly degraded. And this is also why, and this was also already mentioned by Dr. Sundar in detail, many approaches like whole genome sequencing, whole exome sequencing, or typical SNP arrays do not work very well with this degraded DNA. So this is Just an actual example last week, a gel of 4 paraffin extracted DNAs from these tissue blocks. And you can see most of these DNA species are smaller than 220 base pairs. And even if you quantify this by 260, 280 measurement, you will see that not all of this, in fact, is double-stranded DNA, because if you compare this with intercalating dyes like picogreen, you see that in the mean only 1/9 of the DNA is double-stranded. So you may overestimate the amount of functional double-stranded DNA by these assays. So what are the methods that we have as a pathologist? So I think FISH analysis is well established and used in many laboratories. I will compare this with more genome-wide assays like methylation array. I will make some remarks, and molecular inversion profiling. And there is something which works similar, but in a small scale way, is MLPA. So let's look at one of the tumors. So this is ependymoma. They occur in the ventricles or spinal. Here you see this tumor in the fourth ventricle. Here's the cerebellum. And under the microscope, these tumors are very easy for pathologists. So they form this nice rosettes around vessels. They have this round nuclei. And if you see 2 or 3 of these, you can do the diagnosis without being a pathologist. So this is not a problem. The problem is grading. So we grade these as grade 2, still benign, or already malignant, grade 3, but this has no major impact in most of the clinical studies. So as pathologists, we cannot really predict the behavior of the tumors. And therefore, there's really a big need for additional markers, molecular markers, and several have been proposed, but one marker is quite interesting. It has been shown in smaller retrospective studies. It's the gain of chromosome 1q. It occurs in about 20 to 25% of cases, and if it's there, then it's related to worse outcome. These 2 studies have been made by FISH, and as Dr. Sundar already mentioned in the introduction, in our lab and also in many other pathology labs, especially with material which is not well fixed, we have problems to estimate this in all cases. So about 15 to 20% of material does not really result in good evaluation by FISH markers. So we wanted to know if this is really a valid marker in a clinical trial. So we collected more than 200 patients with ependymoma in the German HIT trial, and we decided not to use FISH in all cases, but a DNA-based method. And first, we extracted the DNA, so the recruitment started in 2000, so these DNA samples are more than 13 years old, some of these. And used a method which is also based on ligation of probes. You see there are always 2 adjacent probes ligated to the DNA, which is highly fragmented. So after ligation, after hybridization and ligation of these small gaps, you can amplify the probes by PCR, and then you can do an electrophoresis and identify the specific probes by their lengths, because there are different stuffer sequences in these probes. However, you are restricted to about 20 to 30 probes. So in this assay, we used 9 probes covering the chromosome 1 and some reference probes. And then after electrophoresis, you can quantify the peaks and make statistics on that, and can differentiate between cases which have this 1q gain and which have no 1q gain. And we were able to do this in almost all of the 206 cases. I think there were 2 failures because of low amounts of DNA. And just to show you the outcome, this is submitted. We see that, in fact, also in this study setting of homogeneously treated patients with ependymomas, we see that the 1q gain is a worse prognostic marker, and we were able to show that it's independent of other clinical risk factors, independent, for example, of residual tumor after operation and other important clinical factors. So the problem is so the advantages of MLPA is that you need low amounts of DNA, about 25 to 100 nanogram. You can cover several loci. It's fast, but the disadvantage is that you only see what you're searching for. So you cannot see alterations in other regions. And the most problematic point is that you have to include reference loci. And you have to be sure that the reference loci are not altered in the tumors. So if the reference shows gains or losses, you get really problems with your statistics. So this is the disadvantages. This is why we also moved to molecular inversion profiling. I don't have to introduce it. The principle is similar. You hybridize oligos to your degraded DNA. The trick in molecular inversion profiling is that you get, after ligation, a circularized probe, which is resistant to exonuclease treatment. So you can enrich for the bound probes and remove all of the unbound probes and the degraded DNA, and then you can cleave it again, amplify, and hybridize. The main difference is also that you have more than 300,000 copy informations and not only 20. As already mentioned, you can include somatic variants in that assay. So we did this with the ependymoma cohorts, and again, the success rate was very high, more than 90%, and we were able to see 3 different types of copy number changes. So one type was the so-called structural altered ependymomas. Here's the 1Q that you see here. Also other chromosomes can be altered. And this is the group doing worse. Then we have a balanced group, and we have a numerical group. These are tumors which show gains and losses of whole chromosomes. And when we did a survival calculation, we found that this is really the worst group, and this has been shown several years before with fresh frozen material, because only at that time there was only the conventional CGH available. This is shown before by that group in 2002 that they behave differently. So they are biologically different groups, and this can now also be assessed on paraffin-embedded pathology material. So 1Q gain is clearly a prognostic marker, and it will influence the next clinical studies. I think it's quite clear that this has to be analyzed in front of the treatment. So I would like to switch to the most frequent malignant tumor in children, medulloblastomas. They are highly aggressive, so the symptoms develop very quickly. These children, some of the adults also can get it. These children get brain pressure, cerebellar symptoms. The tumor is always in the cerebellum. Because it's so aggressive, about 1/4 of the patients have already seeding of the tumor cells in the ventricles. The therapy is very aggressive, not only including operation, but also chemo and irradiation, most of the patients. And in the end, about 50% of patients survive. However, the survivors have severe side effects by the treatment of the developing brain and really severe intelligence deficits. So there is a need for more, let's say, specific therapy. The tumor is composed of very immature cells, very quickly growing, and when I started to work in neuropathology and neuropathology research, it was believed that this is one tumor entity. And similar to breast cancer, lymphomas, and so on, it turned out over the years with ours and others' work that in fact this is not true. Medulloblastoma is at least 4 different diseases, developing from different progenitor cells in the cerebellum, using different genetic alterations, leading to different histology and different biological behavior. And I think the belief now is that these different types of medulloblastoma need different treatment. So we are heading towards a risk-adapted treatment. And the next European study, starting in a few weeks in several European countries, will make an assessment of the tumor material just after surgery. In a time frame of 3 weeks, we have to evaluate some of the clinical risk factors as well as molecular risk factors, and then stratify the tumor and the patients to the low-risk group, which will get a reduced treatment to spare side effects, standard risk, and very high risk. And this also relates to the different biology. So one of the groups behaving quite well is a group of medulloblastomas which have an activated beta-catenin Wingness pathway. This is a scheme of the pathway, and it leads to the accumulation of the central component beta-catenin in the nucleus. And we can see this as pathologists. We can show this in histopathology. There is a nuclear accumulation of this protein if the pathway is activated in brown. If it's absent, you only see the counterstaining, so this is a Wingness negative case. So why is this as important? So it was our British colleagues who first identified in a European study called PNET3 that these patients behave very good. You see, very few patients relapse, very few patients die. And this is why it was decided that in a trial it should be tested if these patients still have a good survival with reduced treatment. However, immunostaining may cause problems in some labs. It's not always very consistent, so we search for a second marker confirming the subtype. And interestingly, most of these tumors show also a loss of one allele of chromosome 6. The genomes of these tumors are quite stable, and they have this typical loss of chromosome 6. And this is a molecular inversion profiling. You can detect this easily from formalin-fixed paraffin material. Now, this is the group on the left. So this is a good behaving group which should be picked out to treat the patient differently. The same is also true for another group on the right side, the so-called hedgehog-activated pathway tumors. So these, or some of these, show a more nodular pattern. You see these nodules here. And I think it's about 8 years ago when we really looked at this and showed in 2005, if you just compare cases showing this nodular phenotype to those which do not do it in infants with this disease, it's a clear different survival with the same treatment. And this is why we reduce the treatment in these patients and the survival is still good. So we can spare side effects by identifying these patients. However, histology and sometimes is very complicated. You don't have this pattern in all cases. And the question was, what is the biology of these desmoplastic medulloblastomas? And in fact, it turned out that almost all of these cases have an activation of another pathway, the so-called hedgehog developmental control pathway. This is a scheme of the pathway. Interestingly, these cases have mutations in components of the pathway. For example, this negative regulator called Pietsch, the active regulator called smoothened, or this inhibitory component called suppressor fused. And so you can search for these mutations to identify such cases. However, the Pietsch gene, for example, has 22 exons, so it's a little bit time-consuming, especially in paraffin material. However, these cases show allelic loss. You see, this is an unpublished work on a larger series of these tumors. They either show loss of parts of chromosome 10, where the suppressor fused gene is located, or allelic loss of the long arm of chromosome 9q, where the Pietsch gene is located. If you see something like this, you have also to take into account that in some of these cases, there's a germline situation. So these patients may have germline Pietsch mutations correlating to the so-called Gordon-Goltz tumor predisposition syndrome, and the families have to be offered genetic counseling. So you can see that the loci where these two tumor suppressor components are located are attacked by allelic loss. So I show this once again because I want to show you one detail. This allelic loss frequently is copy neutral. So you see an allelic loss, but the remaining allele is doubled. And this is a frequent phenomenon, especially in embryonic undifferentiated tumors. So if you would analyze these cases by FISH or by array CGH, or by other methods, you would lose the information. You only see it if you can also have the allelic call, which is presented, for example, by this molecular inversion profiling technique, and we use the version number 2 of the OncoScan assay. And then in the end, I want to point out these bad guys, which will be put into the high-risk group. It's believed that these tumors have to be treated more aggressively because most of the patients die with the standard treatment. They look very ugly under the microscope. We call it anaplastic. And this phenotype is strongly related to the amplification of the MYC gene. Now, MYC gene amplification in most labs in pathology is looked at by FISH analysis. And here you can see a beautiful case which shows an N-MYC amplification in the nuclei. In red, there is a centromere probe. This is a hybridization of the same case for c-myc, where you don't see an amplification. And so this is the standard test for this. However, the problem is that it doesn't work so well on the paraffin samples. So the advantage of FISH is that you really get an information on each cell. So I think it's not possible with another method. And especially if there's tumor heterogeneity, you can really count the cells and their copy number. The disadvantage is that you lose information. You do only see what you're looking for. You can look for c-myc and n-myc, but you will lose other alterations. And the other thing is that there's a relatively high dropout rate in standard FFPE tissues in the range between 10 and 20%. And if you want to use it in clinical studies, that's problematic. And the other problem is that if you analyze sections, you also cut your nuclei. And then if you get one signal, you don't know if it's a loss of an allele or if it's a cut nucleus. So you have to correct it by complicated statistics, and this is also something which may cause problems. There's another technology I only want to mention with this slide because we recently published that. You can also use methylation arrays for copy number estimation. If you calculate the copy of the unmethylated as well as the methylated allele, you can derive some copy number information from that, also from paraffin material. However, you would never see copy neutral LOH because, again, you don't have the allelic call with this method. Now, this is now a case of a medulloblastoma with c-myc amplification. You see this high range. You can really calculate the amount of amplified c-myc gene in this case. And there's another structural aberration which is really typical, almost diagnostic in brain tumors for medulloblastomas of this type. They have the isochromosome 17q. You see this here, the 17q is elevated in the copy number analysis, molecular inversion profiling. And these structural changes are frequently coming together. So this is something you can really see very, very easily. So the molecular inversion profiling has some advantages against the other compared methods. It's a genome-wide, high-resolution copy number analysis. You have a high dynamic range in estimating the copy number, especially in amplifications. You see LOH, including copy neutral allelic loss, and you can also retrieve information on those cancer mutations which are put into the assay. You can also see some more fancy alterations of the genome. One very prominent member of this is the chromotripsis, which had been described 2 years ago in the Cell paper. It's an unusual single event in individual chromosomes of the genome, which is scattered in very small pieces, so it's broken in many pieces, which are then rejoined by DNA repair mechanisms, and you get up with a really strange chromosome. This has also been shown in medulloblastomas. Interestingly, in medulloblastomas in patients with germline p53 mutations called Li-Fraumeni syndrome. Here are 2 examples. You see this rearrangement of parts of chromosome 2 and chromosome 12 in such a medulloblastoma. So from our perspective as pathologists, We sometimes see medulloblastomas which look very ugly, so it looks anaplastic, and shows a strong p53 protein accumulation, and this is an indication of a mutation. So normally you don't see this staining. And if you take this case and put it on a molecular inversion profiling, you see that, for example, here the chromosome 13 shows chromotripsis. And if you have this finding, this is a strong indication that there's a defect p53, and this may relate to a Li-Fraumeni situation. So if something like this is found, the patients and their families should be offered genetic counseling, because it's believed that these patients have to be treated differently from normal medulloblastoma patients. You can also use this in differential diagnosis. I didn't mention to you that there are more than 150 different brain tumor entities. It's very complex. There's lots of differential diagnosis, sometimes very complicated by histology only. Here, for example, is a very young child with a big tumor in the cerebellum. You see it here. Here's the rest of the cerebellum. And under the microscope, it looks really ugly, some larger cells, and from purely histology, it's difficult to tell what this is. In fact, it's not a medulloblastoma. If you look at the genome, this was derived from the Nexus software of the OncoScan profile. You see it's quite stable. It's typical for these tumors that they are absolutely stable in their chromosomal content, but they have deletions of chromosome 22. In this case, a homozygous deletion here, which targets the SMARCB1 tumor suppressor gene in chromosome 22. If you stain for the product of this gene, you see that the tumor cells lose this SMARCB1 protein. It's only retained here in the normal endothelial cells and round. So you can confirm this by immunohistochemistry, and in fact this is very typical for so-called teratoid rhabdoid tumors, highly aggressive tumors, which are treated totally different from medulloblastomas. They need a eyes-related sarcoma protocol, so it's very important to identify these patients. Second example, another cerebellar tumor in our reference center, very small undifferentiated cells, so it's hard to tell. Some show these very peculiar multi-layered rosettes, but they can be absent. But if you see something like this, this may be a hint. If you do molecular inversion profiling, you see in almost all of these cases here this amplification of chromosome 19q. It's a microRNA cluster. We have published this a while ago. We have now collected 41 cases of this, and you can see that most of these have this amplicon on chromosome 19 together, or alternatively gain of chromosome 2. It's a very interesting recurrent pattern. If you see this, you can use it as a diagnostic tool. You can verify it by FISH that we've done here. The diagnosis is ependymoblastoma, and this is really a very specific alteration, not present in any other kind of brain tumors. Now, in the end, I would switch back to the adult setting. It's also kind of bridge to the next talk. It's on a diffuse glioma occurring in young adults. It's called oligodendroglioma because the cells look a little bit like normal oligodendrocytes in the brain. So some of these cases show calcifications. The patients frequently develop epilepsy in the beginning. And you can see this interesting pattern. The nuclei seem to be surrounded by clear cytoplasms. If you see this, it helps in the diagnosis, but sometimes it's more complicated and you need additional stainings to make a distinction between oligoid tumors and astrocytic tumors. They are treated differently. Here you see processes in the astrocytic ones which are not as prominent in the oligoid. And interestingly, these oligoid tumors, they have a recurrent alteration. Not all of them, but more than 2/3 of them. They have a co-deletion of chromosome 1p, so the short arm, and the long arm of chromosome 19. And this is an early event, so it occurs both in low but also high-grade oligodendrogliomas. So why does it always seem to be this co-deletion? It turned out some years later that in fact this is an unbalanced translocation shown in these two papers between chromosome 1p and 19q, which leads to the loss of these two regions. Now, why is this important? It turned out in clinical studies that this is a very prominent prognostic marker. These are large cohorts of patients with anaplastic oligodendrogliomas, and you can see here in the Kaplan-Meier survival curves that those patients showing these co-deletions, they have a very long survival with the treatment, and these are the patients lacking this. So you can do this as a prognostic marker. We checked some cases which only had operation without any further treatment, and there it turned out that it's not of relevance. So it's a prognostic marker if treated according to standard protocols. Now, the question is, some of the patients are treated with radiation therapy, others with a combination of radiation and chemo. So what is better? You see that the curves in this first paper are just one on the other. So if the co-deletion is there, the patients seem to do the same way, either if they get both combinations or only radiation therapy. And those which lack this typical alteration do worse. However, that situation changed. So these patients and these studies have been re-evaluated after several years. And this has been presented last year on the ASCO meeting by both groups, and both groups found that after 10 years or even longer, there's a clear survival advantage of those patients having the co-deletion for the chemotherapy. So of those patients having the co-deletion, there was a 14 years median survival versus 7 years if only irradiated. So this leads to the conclusion that this co-deletion in fact is a predictive marker. So if it's there, the patient should get chemotherapy in the beginning. So it should be tested upfront in all of these patients so that the therapy decision can be done on that. There are different methods to evaluate this. So especially in the US, most people do FISH. I have to mention that this early event is a homogeneous event, so you see it in all tumor cells, which makes it more easy. Most US groups use FISH. In Germany and Europe, many groups use microsatellite analysis of 1p and 19q, however, you need the normal blood for that. Some groups, the Dutch groups, uses MLPA. And I think it's very nice also to use genome-wide analysis to really have a picture of the whole chromosome 1 and 19, because it was shown that only partial loss of 1p or 19q or both is not relevant for the prognosis. You have to have the total loss of the short arm 1p and 19q. And this you can see very nicely on genome-wide studies, for example, with molecular inversion profiling, but I guess that Dr. South will address this in more detail. So to summarize my talk is, I wanted to show you, I hope I have convinced you that copy number analysis can significantly contribute to the neuropathological diagnosis of brain tumors. Specific copy number alterations that I mentioned, the 1p19q story as well as the 1q story and ependymomas, have prognostic and even predictive impact. That FISH as a method can give you copy number information on a single cell level. However, there's quite a high failure rate in FFPE tissues. That the DNA derived from these old paraffin blocks can be used for copy number analysis by molecular inversion profiling, and this technique is suitable for high-resolution genome-wide copy number analysis, including the detection of copy-neutral allelic loss. With this, I would like to thank you and hand over back to Dr. Sundar. Thank you very much.